Hello, everyone. My name is Hosein Azarbonyad. I am a machine learning scientist in Elsevier, and today I will talk about one of our ongoing initiatives with Elsevier and some universities in the Netherlands, which is on annotating and indexing scientific articles with rare diseases. This is a joint work with Zubair, Max, and George from Elsevier and Rick from Erasmus Medical Center. But before diving into the details of the project, let me briefly tell you about what we as machine learning scientists or data scientists do in Elsevier. So our goal is to use new capabilities and technologies in the field of machine learning, NLP, and artificial intelligence in general to increase our content utility. So basically, our content are the articles published by Elsevier or are collected by Elsevier. And this content is usually in an unstructured form because most of the time the content are the articles published by Elsevier, which are in textual format, and we want to use this machine learning techniques to turn this unstructured content into structured content, which is easily accessible and readable to the users, and we try to extract insights from this content that can help users to understand the articles or a research area they are interested in. And most of the times, the content, as I said, are in textual format. So we use a lot of text mining techniques, but also we have images and videos attached to the article. So to process them and to extract information from such content, we use image and video processing techniques. We do have a large collection, two large collections of articles. The first one we have is Science Direct, which contains all the articles published by Elsevier, which is about 80 million articles, and we also have Scopus, which is a collection of articles published by Elsevier and also other publishers. And this database contains about 80 million articles. So as data scientists, we deal with a large volume of unstructured data in Elsevier. Now, let's dive into this particular project, which is, as I said, on indexing articles with rare diseases concepts. But why we are interested in indexing articles with rare disease concepts? So in only Europe, about 30 million people are suffering from a rare or orphan disease. And the disease considered to be rare if it occurs in less than 1 per 2,000 people. This number is actually quite a high number, but obviously rare disease patients are entitled to best possible care. But to find the best possible care, you need to know what are the excellence centers around different kind of diseases and what are the key research initiatives for the various different rare diseases and who are doing this kind of activities. One way to answer such questions is to perform a deep bibliometrical or scientometrical analysis to understand what are the most active research centers around different rare diseases by looking into the publications they have published over the years. And that's why exactly we are interested in this project. So our goal in this project is to map research outputs, which are articles in Scopus, to concepts in OrphanNet, which is an orphan disease taxonomy. I'll explain it later, to track research on rare diseases. So basically, we want to find out, we want to build a system that can detect what articles are related to what diseases. And we want to provide different insights and analysis on top of these outputs so that people or patients can easily see what are the most active medical centers or research institutes on different rare diseases. Apart from the application I just explained, medical centers can also use this output, for example, to showcase their research output to be recognized as an expert center on particular rare diseases they are working on, or to get funding in research areas or rare diseases they are expert at, or they can use this output to recruit, retain, and promote talented researchers and faculty members. But indexing articles or annotating article with rare diseases concept is quite a challenging task. I will here list a few challenges we are dealing with. We have tried to address some of these challenges, but most of these challenges are actually open challenges. The first one is that there is no label articles with rare disease concepts to train or evaluate any supervised model. And also the expertise on rare disease is also rare. So it's really difficult to find people who can help us with building a large collection of label articles so that we can build models that can learn from those articles. And some rare diseases are only rare in a specific part of population. For example, inflammatory bowel diseases is rare in children, but not so rare in adults. So any system that you want to build to annotate articles with rare diseases should be able to pick up contextual information from the articles other than, for example, the names of diseases. And some of the rare diseases are very similar conceptually or synthetically, and their differences are very difficult to recognize. For example, here we see two diseases that are basically very similar, and any system that you want to build should be able to recognize this kind of little differences in the names of diseases. And sometimes these even the symptoms or the genes causing diseases, they are also very similar, and any like shallow model that you want to build would fail to detect the differences between different rare diseases. So you need to build a model that can understand the context and also understand the genes and symptoms and can differentiate between them. And the orphanet taxonomy, the taxonomy we use, as any taxonomy might be incomplete in certain areas. So this taxonomy is being updated very frequently, actually. So any system that you build should be able to cope with these updates and should be able to apply such changes in the taxonomy structure. And there are also some classical challenges associated with any taxonomy, such as polysemy and synonymy. These challenges also apply to orphanet taxonomy. So we need to deal with such kind of challenges as well. And as I said, most of these challenges are open challenges, and we don't have a good solution for most of them, but we are trying to build something that is effective enough and look back at these challenges and try to address them at the later stages. And to index articles with rare diseases, as I said, we focus on the collection of articles we have, which is a Scopus. And on the other hand, we use rare disease taxonomy, orphanet, to basically track different outputs on different rare diseases. Our collection of scientific articles is basically a Scopus database. We only focus on the publications in the past 10 years. This is in line with how different research centers in Netherlands, at least, are evaluated to be considered as an expert centers. For evaluating research centers, the criteria is to look at their the publications published in the past 10 years. So we are trying to kind of mimic that in our analysis as well. This subset of articles contains 36 million records with a rich set of metadata that I'll show later on. And orphanet is a structured vocabulary for rare diseases, capturing relationship between diseases, genes, and other relevant features. It contains about 9,000 concepts, which are organized in a hierarchy. And as I mentioned earlier, it's updated monthly, and it follows the standard guidelines on deprecation of terms in taxonomies. This is our annotation or indexing engine, basically the pipeline, which is quite a simple pipeline. As I said, we start with a Scopus database. From Scopus database, we apply different kind of filters. For example, we filter on the year to track only the publications in the past 10 years, but we also filter based on organization or country to see what kind of organization are most active in what kind of diseases, or what diseases are frequently studied in what countries. From Scopus database, we only select title and abstract and keywords of articles to perform the matching with diseases. We haven't used we actually tried to use the full text of articles, but it turned out that it leads to a lot of noise, which are false positives. And so we focus on these three pieces of information: title, abstract, and keywords, and are trying to find ways to deal with the noise in the full text, so that we can also use the full text of articles. So we concatenate these three fields to build our documents. Then we feed these documents to our annotation engine. And from the orphanet taxonomy, we also use the names of diseases, synonyms for the diseases, and the genes associated with different diseases. We feed this information to our annotation engine, which does the matching between the orphanet concepts and the documents. And as the annotation engine, we use SciByte's termite annotation tool, which is a named entity recognition tool that rapidly scans and semantically annotates raw text with entities from over 50 biopharma and biomedical topics. We use this system because it's very effective and very fast. Later on, I will show the performance of this annotation engine. But here you see an example of the annotations done by this engine. As you can see, it tries to find the names of diseases, but also the synonyms of the diseases are also matched in the text. A few words about this annotation engine. So termite covers only, it covers most of the orphanet taxonomy, about 98% of the taxonomy. But the other 2% is not covered because of the different like frequent updates applied on top of its taxonomy. And to cover the other 2%, we use a string matching, a simple string matching approach, which matches disease names and synonyms with the document. So basically, it looks for the exact mentions of the names or synonyms of diseases in the documents. Termite performs works a little bit in a more advanced way. It in addition to the exact matching, it also does the fuzzy matching between the synonyms and the names of diseases, and it also tries to disambiguate detected diseases for a document. So, for example, if we have a mention that might be a mention of two different diseases, then it tries to find the one that is the most relevant or most applicable in the context. To do so, it tries to find to use additional relevant ontologies such as gene disease relations. This information is also included in the orphanet taxonomy, or tries to use common entity disambiguation techniques such as the relationship between detected entities. So this is our annotation engine, but we would like to see how effective it is. So we want to see how good this approach is in annotating articles with rare disease concept. Well, the evaluation is particularly difficult for this task because, as I said, there is no annotated data set, and the domain expertise on rare diseases is required for annotation, which is really difficult to find. And as an alternative, we tried to look over other well-known taxonomies that have labeled data sets, and try to see if we can use those taxonomies and their associated annotated articles to evaluate our approach. But it turned out that these famous well-known taxonomies only cover a small portion of the orphanet taxonomy, so we decided not to use them. But we still wanted to see how good this termite engine is in annotating our articles with rare diseases. So we designed a very simple and very basic evaluation pipeline. So what we did, we focused only on the publications published in the Netherlands, and we used this engine to annotate these publications. Then for 4 big medical centers in the Netherlands, we sent them their index articles, and we asked them to manually inspect which approach performed better. And as an alternative to the termite engine, we also have the string matching engine. So we sent the annotated articles to these 4 big medical centers in the Netherlands, and we asked them to let us know which engine assigns better rare diseases to their published articles. And here you see the result, the number of wins of termite and the number of wins of string matching and the number of ties, meaning that the diseases associated by these 2 approaches were exactly the same. It turns out that the termite has a higher annotation performance compared to the string matching for 3 out of 4 medical centers. For the Amsterdam medical centers, actually string matching performed better, and the reason is that we have tried to adjust termite in a way that it has a higher recall, but for this particular medical center, they cared more about the precision. And as a follow-up step, we would like to make this pipeline more adjustable so that the users can opt for precision or recall or any other metric that they would like to optimize. And for many articles, the disease assigned by termite and string matching are the same. And the reason is that the orpha concepts or rare diseases tend to be very unique and specific, which are detectable by string matching in our articles as well. Here you see the statistics of Scopus publications on rare diseases detected by the pipeline I described. And well, here non-unique means that refers to the number of the matches between a rare disease and an article, and unique means the unique number of articles. So there might be multiple diseases assigned to one article, and the non-uniqueness reflects that. And so in total, there were about 2.4 million articles on rare diseases in the past 10 years in Scopus, and about 66,000 in the Netherlands and 647,000 in the EU. So we are in the process of building a dashboard and building like a standalone analytical tool on top of this data, so that people and researcher, patients, and scientists can track different kind of publications on different rare diseases. But as I said, we are in the process of building this, but as an intermediate step, we imported this data into SciVal, which is another product of Elsevier, and I just want to show a dashboard that we have there. But again, I want to let you know that this is not going to be the final product. The final product will be a standalone product outside SciVal. So here you see a dashboard we built for a particular disease, Duchenne and Becker muscular dystrophy. What you can see is the number of outputs or number of publications around this particular rare disease. You see about like exactly 308 publications on this disease in the past 10 years, 1.7 thousand authors and different kind of metrics. If we go to the collaboration part, you can see the different kind of collaborations on this particular rare disease. You see a big portion is only national collaboration, and there are some international collaboration and some institutional collaboration, and only a small portion of the articles published on this particular rare disease are academic or corporate collaboration. Moving on, we can track different kind the number of publications over the years as well. As you can see, the number of publications are growing, but this particular drop is because we haven't included all articles published in 2021. And moving on to probably the most important thing for users to see is at least the institutions who are most active in this particular research output. As you can see, this particular university from France is the most active one, then University of Twente from Netherlands, and so on. Also, you can see the authors who published the most in this particular rare disease. So basically, this is similar to what we have in mind to build as the final product, but as I said, this will be built hopefully soon, and this will be a standalone product. Now, well, to conclude, I just explained how the different kind of challenges associated with indexing articles with rare diseases, challenges such as lack of large enough labeled data and rareness of diseases in particular part of population, and the conceptually similar diseases. We have tried to address some of these challenges, but most of these challenges are open challenges. We have used termite that can address some of these challenges, but in combination with some entity disambiguation techniques, and I just showed how the combination of termite and Scopus can result in a rich data set of scientific articles with rare diseases, and how what kind of insights it can provide for the users. And I also, as I said, we are only using a very small portion of articles to perform the matching between articles and rare diseases. We haven't used the full text, for example, because of the noise, but as a future work, I think we need to work on trying and try to find ways to deal with such noises to increase our recall. That's all I wanted to say. I hope you enjoyed this presentation. If you have any questions, please let me know. Here you can see my email address and also my Twitter account.